Hello, everybody. We're going to get started with this next session. I'd like to introduce Laura Guillardin. She's going to talk about continuous cover forestry and genetic diversity, a case study of England's Douglas-fir and western red cedar plantations.
Okay. Hello, everyone. I'm Laura. I'm a fourth-year PhD student. And yeah, I'm going to talk about the genetic diversity in planted UK conifers managed under continuous cover forestry. This is the outline of the talk. And first of all, what is continuous cover forestry? So in the UK, of course, we have, we have plantations for timber production, but it's being advised to switch into a continuous cover forestry. I don't think it's the whole screen there. Okay, that's fine. Thank you. So basically, what continuous cover forestry is an approach to manage the forest that aims to increase the diversity of species and to get to an irregular structure while maintaining the timber production, but adding other forest services. Why is this being advised? Well, under the climate change context, these forests are meant to be more resilient to pest and diseases outbreaks, climate, high temperatures, or extreme temperatures, and droughts, and all this that we know. So there is a lot of knowledge and literature about how to transform from a plantation to a continuous cover forestry or irregular stand. There is also a way to track how this transformation is going by looking at these profiles of number of trees per diametrical class. So this will be a plantation sort of profile. And here we can see the reverse J of a more irregular stand. But what we are interested in is in the genetic makeup of these planted trees, because, of course, this will lead into the actual potential adaptation to climate change. And because the continuous cover forestry approach will relay natural regeneration, we want to compare between the adult and the natural region to see how the genetic variation is maintained. So this is the main question of my project. The species that I'm looking at, you're very familiar with, so Douglas-fir and Tuya plicator. They're two of the preferred planted species in the UK for timber production. And from now on, if we see this silhouette or this one, I'm referring to Douglas-fir or to Tuya plicator in this case. The sites where I'm doing my project, they are in the south of England. And I have 5 sites, and within the sites, I have 8 stands. So these stands can be just one species or 2 species within the same stand. The first thing I did was to perform a forest survey or an inventory to understand how these stands look like. So these are the values of trees per hectare for each of the sites and the total number of trees. The next thing we wanted to see is the number of trees per diametrical class. And we put it in the order that kind of matches the diagram of transformation into continuous cover forestry. So these are our 8 stands, in green Douglas-fir and in blue Tuya plicator. So we can see how we have from early stage of transformation to late stage of transformation. We also did an estimation of the species diversity on these stands, and we can, we can see the pattern follows the same trend than the stage of transformation. We also wanted to see the same, but for regeneration and shrubs. And in this case, we didn't find the same pattern. And actually, two of the kind of early stage of transformation show high levels of region diversity and shrubs diversity. Just as a quick example, this is how an early stage plantation would look like. An intermediate plantation would look like with more region around and smaller trees. And this will be the late stage of continuous cover forestry that we have as an example in the UK. So how I did this, how I looked at the genetic makeup for both species, it was quite similar with some difference that I will point out later. But first of all, it was the sampling. So we sampled 50 adults and 50 juveniles per site. The adults, we needed to use a big slingshot to get over the branches to get some samples down. And if we play the video, we can see the technique, very delicate technique. There were many ways of doing this, and sometimes we got to branches over 20, 25 meters, and sometimes we were getting just a bit of needles, and sometimes we got more than enough. We were always shocked about that. Well, so after that, the DNA isolation, which if you have seen the poster session, that's where I'm talking about that. So the next step would be the SNP set design and the genotyping. For Douglas-fir, we use a public data set of SNPs that was available, and we filter through different criteria, and then we genotyped our stands using two platforms. We use Sequenome, an outside platform, and we also use Fluidium, our own genotyping system. And if anyone is interested in the high throughput of this genotyping system, we can talk about it later if needed. That was for Douglas-fir, but for Thuja plicata, we didn't have any available SNPs at the moment. So we performed our own genotyping by sequencing step to perform a SNP discovery step. SNP discovery. Is this a problem with the micro, or is it just me? Sorry. Okay. A SNP discovery step, and we also got a big set of SNPs, and from here is very similar to Douglas-fir. We performed the same filtering to get the number of SNPs that we were looking for to then genotype our stands. So the results, we look at different genetic diversity indicators, and the first one is the heterozygosity. So on the left, we can see that overall, all the sites of Douglas-fir show high levels of genetic diversity. There's a significant difference between these two sites, but overall, they were all really, really high, as we expected. This also follows all the results from other projects in Europe. For Thuja plicata, 3 of the sites showed high levels of heterozygosity, but long-read, I don't think you can see this very well, but is around 0.24 heterozygosity, which is the one that is the minimum, and this sort of level of heterozygosity could lead to genetic drift and inbreeding depression. But what we were interested on was in comparing these two groups, the adults and the juveniles. when we look at the differences between those in Douglas-fir, there's no difference. And when we look at this same thing in Tuya plicata, we found that for long-legged, the adults show higher levels of heterozygosity than the juveniles, and the opposite in Stourhead. Then we looked at the effective population size. So I'm not going to introduce this because it's been talked before and very well explained, but I just want to point out that we took into account the minimum seed orchards NE as 10, and then we also have the 50-500 rule of short time minimizing breeding, and for effective population size higher than 500 to maintain the genetic variance in the long term. And the results for Douglas-fir, all the stands and all the groups are over 50. Only just one of the groups are over 500, and is the adults in Greenwood. But if we look at Tuya plicata, note that the scale is very different here. So we cannot even see the 500 line. All the sites and the groups are under 500. Only 2 of them are over 50, and some of them are even under 10. The last genetic diversity indicator we wanted to look at is the inbreeding coefficient. And in Douglas-fir, we didn't see any sign of inbreeding, while for Tuya plicata, as we kind of expected, we saw the inbreeding from more or less every stand. And even though there were no significant differences, for 3 of the sites, the inbreeding coefficient was higher on the juveniles than in the adults. And these may not be significant now, but maybe in the future could be a difficulty. And just to wrap up, if you remember this slide from before, this is the species diversity. And here I just want to point out that this stand, which shows the highest species diversity, and is the one that is in the latest stage of transformation, but it happens to not have the best effective population size or other genetic diversity indicators. Actually, Tuya plicata effective population size were under 10 in some of the, well, in one of the groups, and for Douglas-fir, it was under 500. So here I just want to emphasize that although a plantation can be naturalized and we can increase the diversity of the species and the structure, it could be like the aesthetic success that we were talking at the beginning of the day in the first keynote, that it might look like it's fine, but I think, and I want to point out here, that we can't assume that this is an adapted natural population. So more genomics and genetic studies should be done if we want to transform these plantations to more naturalized forests and relying in natural regeneration, because in the future it could lead to inbreeding depression and lack of potential to climate change pressures. And that's it. I want to thank my field assistants, the lab members that came with me to the field as well, my lab assistants, of course my supervisor John Mackay, co-supervisor Gary Kerr, the current and past group members, the lab technician Patty and Sarah Rogers, and the owners of the forest that I'm doing my project. And yep, thank you very much.
Thank you so much, Laura. Any questions? We have time for a question. Anybody? Oh, we have a couple. Good.
Hi, thanks for that. I was wondering if there was anything known about the seed sources for these various, I assume, were these originally all plantations that have been converted over time?
So there are different cases, but that's a great question. Of course, some of the sites that we were working in were, as you said, very old planted, like in the 1930s, so there are no records of them. Some of them are newer, but even though we ask for the records, sometimes we don't get very precise answers. And what we want to do, and that's something that is ongoing, we have access to some provenance trials data, so we want to kind of analyze that, some individuals from those to see if they, combine these results and do some genetic structure with our DNA, that provenance trials, to see if we can understand where they come from. So, but that's something we're looking at.
It's okay, John stole my question, so you get a free pass on this one.
Thank you.
Okay, thank you, Laura. It is my pleasure now to introduce Melissa Spearing, and the title of her talk is "What Does the Future Hold? Aiming the National Tree Seed Center's Research and Conservation Strategy Towards 2030 and Beyond." There you go, Melissa.
Testing, testing, 1, 2, 3. That's Victoria's line. Yeah, so I'm grateful to be here today, and the CFGA has always been one of those amazing places for talking to people, networking, and just being involved in a great organization. So Dave and I had a really great workshop with the Tree Seed Working Group yesterday, so this is a little bit of building on that, but also just all the great talks that have been here. So I'm really here today presenting on behalf of my management team at the Atlantic Forestry Center. So we're based in the unceded traditional territory of the Wolastoqey First Nation, which is known as Fredericton, New Brunswick. So for those from out of country, we're down in this little province. And yeah, so this is just my main objective, really, is just to inform you of some strategic work we've been doing and some new priorities for the next decade that's going to sort of maybe lean on some of you guys at certain times. And looking a lot into the crystal ball is always difficult. If you're a seed banker, Sean O'Ben's talk was so great to follow because it's the kind of work we're trying to do with so many different people across Canada. So I think we're all fairly well aware of the fact that beyond forest industry, that climate change really is having an impact on all of our forested landscapes. So even if we reach net zero tomorrow, a lot of the foundations of sustainable forest management are pretty worrisome to consider that they're going to be stable. So it's also pretty worrisome for us in northern countries that, Canada in particular, and the boreal forest is warming at twice the global average. So this main graph here of the cumulative effects and resiliency of forested landscapes as intact ecosystems and functioning ecosystems and the services they provide for us, it's from Natural Resources Canada produced a new research agenda for the next decade to try and address cumulative effects to all these landscapes. So beyond forest industry that we've always supported, it includes preparing our facility, our mandate for new research, new models, new tools and solutions for things like ecological restoration objectives, new traits, industrial reclamation on very challenging sites that most of us foresters don't thankfully have to deal with all the time, and more forest pests, pathogens, floods, extreme climate. So yeah, we're really just here to try and say that there's a lot of things at seed conservation. It might just be one tool, but for sure if we prepare for it, if we're collecting down before these impacts happen, we hopefully, as has been explained now, is that we don't wind up with really small populations that are effectively not useful to us in the future. So for those that don't know about the National Tree Seed Center, I know a lot of countries have these institutions. Our organization's been around since the 1960s, and we've been part of the Petawawa National Forestry Research Institute's tree breeding program. So we were really put in place to store seed, obtain it, help keep it alive as long as possible so that tree breeding programs could get going. And of course, it's so successful today across Canada that most of the provinces and territories now really have, you guys are shining examples of that, the fact that it's operational, it's useful, it's in use, and it really is making an impact now on wood supply and forest genetics and conservation. So in 1966, our seed bank actually moved, and it kind of coincided just afterwards with the signing on the Convention on Biological Diversity in 1992. So that was the point where we started branching more into non-commercial species and really looking at technology, building on things like somatic embryogenesis that was happening at that lab then with Dr. Jan Bonga, but also starting to work more on the new molecular tools that were coming along. So we still primarily conserve mostly seed, but we're also now prioritizing more native species and populations that are under threat. Especially in the southern settled part of Canada, there's a lot of species that have pretty small effective population sizes. So we're also now still helping to support a lot of national efforts in growing and maintaining forests because we know that's going to be an effective solution for climate change. So I'd also just like to point out too that we're doing right now a lot more, we're being called on to do a lot of knowledge exchange. People want to know how we've done what we've done for so long because they want to start their own little program in their own community. And I think that's such an important thing that we're playing a role in. So wherever we can, it's not just about collecting things in our little stamp collection, it's trying to help people that want to do this in their own backyards. And I'll just point out too that we're the only national program that has a mandate for this, and often we've been inheriting things that may have been put away in a freezer that someone forgot about, and we're inheriting a lot of that material to try and keep it available. So we've built up a pretty impressive living library, as we like to call it, and I call it the most biodiverse time-traveling forest machine we have in Canada. So this is just a summary of all of our seed collection activities, so the number of collections that we've collected ourselves with our own staff, registered or had donated to us each year, and then a running total of sort of the initial number of seedlings that we started with. So it's not a massive bank with 250 million, but that's just what's available from our website. And we have seed lots from a few different places in the world, but we have really good coverage of a lot of species in Canada. Our number one is white spruce. We still have almost 1,600 collections of that available today. So, we still provide a lot of seed for research and education. So this is Fleming College they order seed from us every year to do their propagation labs with their students. So their forest techs know where their seeds come from, and they were super happy this year because it all came from within 100 kilometers of the college. They thought that was pretty neat. So yeah, our oldest seed lots are from the 1940s. We still have a 1948 Norway spruce seed lot we still test. We've got almost 31,000 or 32,000 germination testing records. We've been expanding our seed science and technology as much as we can. And we've provided seed to over 70 countries, though most of our seed goes to Canadian research programs. And of course, one of the things that I like to point out is there was a bit of a lull in activity at this point, but in the last 4 years, just before COVID, 2019 was our largest seed collection year in quite a long time. And it's mostly because Donnie, at the last CFGA meeting, made the mistake of talking to CBC right before he got on stage, and then he spawned a massive national collection effort for ash. So black ash is actually our number one hardwood species now in our freezer, and we've been able already to capitalize on that for some projects with the US. So what's going to fuel our new strategies? This is just one of the biggest things for us is a big uptick in number of staff we have. So we went from having one technician in our lab in 2019 to having 14, and we're hiring 3 more this summer, which is great, and then we're trying to emphasize hiring Indigenous folks. So this is just a bunch of our lab techs doing seed testing. So our operations, obviously our bread and butter is still collecting, processing, storing, and testing seed every year. We do almost 900 germination tests a year on average lately, and we've added some X-ray machine technology. We've been investing more in non-destructive testing so that we don't have to destroy seed samples for that, especially for valuable research. So we ship out seed orders again. We said we supply about 100 seed lots a year, and last year we supplied 76 different species and over 570 seed lots. So we're also doing a lot of documentation right from the day that it gets collected. We're doing a lot more work now on making that data accessible. We're trying to get more information into things like the Q-Seed Information Database topic. Some of these ones where seed trait data and modeling is becoming very useful. So a lot of the records we have, we never throw a piece of paper away, which is good and bad some days. But we've definitely kept as much as we can because we know it might be valuable if we want to go resample. We're also, managing and trying to optimize our operational processes. We learn with all the provinces. We learn from different countries how they're doing things. We try to keep innovating our processes with new tools, and we really encourage researchers and students to come to us if they want to test out an idea, try new technology. We learn with everybody that we even provide seed to. And we're just, the former manager and people within our organization for so many decades, they were really on the leading edge of seed conservation research, trying to come up with new protocols, how to test species, how to collect them better, how to make sure that the material we have, that we can maximize its use. And when we have clients that are trying to grow things out, try to give them the best advice that we can. So we're always trying to conduct ongoing experiments. And anybody in our lab that has an idea, if they have time in their schedule, we're trying to make that, encourage them to do their own little research projects so they learn. Learn by doing. So yeah, at this point, a lot of that information that we've gathered, it's obviously feeding into what we think we can do, and it being a realistic plan, and obviously the strategy, if it's successful, we're going to generate lots more collaborations. So our vision is that we're always trying to, but really still want to maintain to be a national center of expertise so that people come to us for questions and advice, and we can direct them to local experts if we need to, but we're trying to be that point of contact. We want to play a key national role in conservation, education, science, make sure that seed collection, conservation is something that foresters hear about, they know that this is really still an important piece of adapting as the new challenges come at us. And we're really still trying to contribute, obviously, to CFS's efforts to preserve the biodiversity. We do a lot of national reporting on this, so all the metrics that we want to try and stop biodiversity loss, this is one tool, but it's something that we do. So we also want to do anything we can to meet the climate change objectives. And there's 4 new priorities that we're going to be doing. So some of you have heard about the 2 billion tree program. We've been heavily involved with that right from the very beginning, and even anticipating certain things beforehand. So we're really working with a lot of the capacity building stream and proponents that have gotten money, try and help them make that happen and do it right. We have a huge new program on supporting Indigenous leadership and inclusion in seed conservation. Historically, they've been included in it, but we're really trying to increase that knowledge and exchange. We've always had a national role, but really trying to revitalize it and get feedback from people that we're doing as much across the country work as we can. And we're going to keep working towards a national collection strategy. So even though we have a lot of stuff in our bank, there's still a lot of species we can advance on. So for 2 billion tree programs, just some things that we would like to do is that we're going to keep mobilizing the information we have. We want to be supporting the seed supply producers, particularly those that may not have some experts to lean on. So we've been, again, engaging with a lot of 2BT proponents. We've been developing and sharing knowledge products, and we're really trying to emphasize low cost, low tech, low barrier solutions and equipment for people that are trying to do things locally. Not everyone can use some of the provincial seed centers, and we're really trying to help fill that gap a little bit. So we've been producing things like a YouTube video series, which is kind of a big deal for us because it's really hard to sometimes to get, make it look fancy and behind the scenes, and our staff are getting very, very comfortable behind a camera. So yeah, just some of you can take some tour of this. I can share this if you like. And of course, one of the big things for us that we've been always trying to do is support the tree seed working group. So I help out with publishing, try and encourage people to share information. And for us, that's a great way. Our staff, when we produce it, they like reading it. So it's good to get our staff to write in as well. So just anyone that might have visited our seed center at the last CFGA when it was in Fredericton, all of this equipment was put into a tiny little closet room, and we got to finally take over some space. So we've been definitely investing in more equipment, testing things out with people. We encourage a lot of folks that are getting involved with 2 billion, if they're seed collecting for the first time, come to our lab. You can bring your samples. We'll clean it with you, show you how to do it hands-on. And one of the things behind the scenes we're really working to do, and this is a big personal effort of mine, is trying to improve some of the data flow and get our data out there, easy to find, downloadable, accessible. Our website right now, the system's a little old, so it's difficult sometimes for clients to order from us. We're going to try and fix that and just explore more partnerships on how to account for the fact that if there is seed somewhere else, right now it's a lot of manual referring people to where there might be a little bit of surplus seed that we might not have, but figure out if there's a way to make that simpler. So we want to be supporting Indigenous leadership and inclusion in seed conservation. So it's part of $2 billion as well, but there's more funding coming as well for them to be involved, even for Indigenous protected conservation areas. So we've been engaging with a lot of communities, developing products and knowledge products for them, and we've got a whole team of people working on a national working group to do seed conservation in that realm. So some of you may have been aware of the fact that my boss got the ability to take his RV and his wife and drive all the way across Canada and meet with communities all over this blue line. I was in Southern Ontario doing some. We also sent staff to most of the Maritimes and Newfoundland last year. So that really got us in touch with about 75 different communities and generating a lot of interest of forest plants and non-timber forest products in particular of interest to them. They're very interested in restoring medicinal harvesting areas, restoring plants that depend on the forest but aren't necessarily part of any other program in Canada right now. So that trip really led to a lot of communities wanting to come to us. So people are sending us their Indigenous guardians, their technicians, and they're bringing their seed samples again to us to train and learn. And for us on the receiving side, the important thing for us now is relationship building and listening to what they have to say and incorporating it into what we do. So when we're collecting now and we're trying to look at our lab processes, we're trying to do this whole two-eyed seeing thing and trying to figure out if there's things that we can adapt that will better respect the material that we're starting to work with them on. So we're doing a lot of recording Indigenous knowledge with our technicians, updating our training to include that. And there's an engagement toolkit we'll have that'll be specific to what we've learned so far. And it's just been really fun. It's been fantastic sessions. These things have been, we've led 3 of them, so it's been great. And so this part is some of what I do in my role, but trying to strengthen some seed science and seed collection within the Canadian Forest Service itself, trying to have more of our technicians in different offices, because we have 5 different offices across the country, helping with seed collection. They're out doing other field work in pathology or other field work, and we're really trying to get them to look, again, with 2 eyes and see if there's trees or cone crops we might be able to get, other crops. But there's a lot of CFS researchers that are having a renewed interest in looking at our bank, and we really want to build that up so that there's some really active seed science, again, coming out of the CFS. So just a quick map of our locations. And at the international, national levels, getting back involved with some of the groups, such as ISTA and the OECD. We've been talking quite a lot now with different US organizations, trying to anticipate some of the seed collections we might need for research from that respect in terms of assisted migration and near-native species that we might trial in Canada. And the number 4th priority would be a targeted national collection strategy. This has kind of always been what we've been trying to work at, but really would like to get more support for being much more proactive, much more regimented against the threats that are coming. So we're going to try and do this, obviously, in connection with everyone who is already an expert. We are doing a lot more partnership now with Parks Canada and trying to use a lot of new technologies, like a naturalist might be one thing, but we're trying to find other ways to facilitate cone collection opportunities or crop collection opportunities. We've also been working a lot more with the CFIA in terms of being proactive on pest threats. And this is just a quick recap of some of the quantitative targets that we tend to have and will boost. But for every species, if we can do some things like Sean's work in finding species-specific targets, we'll try and work at that. So there used to be a group called Confergen, or is a group called Confergen, and that's been kind of the group we've leaned on to try and anticipate forest health threats before they occur. So one of the other things that's exciting for us is the fact that we can build on Martin Williams has talked about butternut today. We really do want to expand on that success. So our cryopreservation lab is going to get larger. We've sent technicians to Valerie Pence and Christina Walters' lab this winter so they could learn more cryopreservation techniques. And we're really engaging with a lot of the global conservation consortia groups for really challenging and difficult species. So, our founding principles, our programs, our facilities were always designed with long-term forest genetic goals in mind. And I know some people have heard the slogan now, right tree, right place, right time. And I do still think, though, it boils right back to one of our founding visions here that Kit Yeatman always said. So one of the most important things we'll always say, that seed origin being first, last, and always, is that we're going to hold true to that. And that if you or your organizations help contribute to these jars of seed, thank you. We're trying to keep them as long as possible. And if you've ordered seeds from us, we always want to get results back and let us know how they did. And if you're interested as a student in anything here, we know there's still a lot of stories inside these jars. We just haven't figured out what they have to say yet. So that's just quick from us. And this is our team at the Seed Center last year. And if you'd like to talk strategically at that level, you can talk to Donnie or Lucy, and I can answer a couple of questions that are on the technical end. But thank you very much.
I wonder if you have any priorities for what population target to include in your collections, in the sense that with climatic change, what Hungarian researcher Csaba Matthias called the exotic limit, meaning the low altitudinal population or the southern population of the distribution of each species are the ones that are in more danger. So do you have some priorities to collect first those populations? I wonder.
Yes. We've had, in our database right now, our priorities have been what was prioritized the last time in 2012 from the Confergen group. So there's been national jurisdictional surveys, and all the provincial groups have put in and said this was sort of what they wanted us to work on. So we're still using that ranking system. We've been evaluating whether we would adjust it with some of the new techniques that are coming to light. But essentially, I mean, we do have I can produce at any point right now a map of where we have almost no collections for a certain species. We're really concerned now, especially with the fact that I can use our germination testing data. I want to make sure that we are replacing a lot of declining seed lots where there's still big populations, but we are trying to sample across the range for a lot of species. And there are a lot of eco-districts. We're using the federal eco-district system right now just for planning and spatial, spatial differences. But at any point, I can give a collaborator a map that says you're in an area where we have nothing, and we really would like some. Our minimum is 15 per eco-district right now, just as a base for most species. But we've been really trying to react lately to things like hemlock woolly delgid, emerald ash borer. Those have been the two that we've spent the most amount of time on the last 2 years, just to get things before the trees and populations. We know that the mortality rate's so high. But yeah, if there's at any point somebody's interested in that, I can make a map.
Any other questions for Melissa? No, let's give her a big round of applause. Thank you very much, everyone, for staying right till the end. We got about 25 minutes before we load the buses, so take a breather. Thanks.